Welcome to this video on reaction-diffusion systems for 3D printed artificial coral. We'll explore biomimetic morphogenesis for marine restoration ecology. Artificial reefs currently rely on simple geometries like concrete blocks, which lack the biological complexity essential for thriving marine ecosystems. Natural coral surfaces, however, maximize turbulence, nutrient flow, and larval settlement, leading to high biodiversity. Our hypothesis is that biomimetic geometries, generated using reaction-diffusion equations, can dramatically improve reef colonization success. Natural brain coral morphology provides an optimal design, featuring intricate, maze-like patterns. This complex geometry is highly organized and optimized for efficient fluid transport and maximizing surface area for symbiotic algae. Intriguingly, these patterns strongly resemble the results of Turing reaction-diffusion morphogenesis. A reaction-diffusion equation describes how two substances, an activator and an inhibitor, diffuse and react to form complex spatial patterns. The general mathematical form involves terms for diffusion and reaction kinetics that govern how these two interacting chemicals spread and influence each other over time, ultimately generating stable, self-organized patterns. Segmentation from reactive diffusion systems is central to our approach. D2L, or diffusion to lumped or discrete models, is a multifunctional architecture paradigm that translates complex reactive diffusion outputs into nature's designs. Turing pattern formation, proposed by Alan Turing in 1952, explains how uniform states break symmetry. The process involves three steps: activation, where a chemical increases its own production; fast inhibition, where an inhibitor suppresses the activator and diffuses quickly; and finally, emergence, resulting in patterns like spots, stripes, or the mazes seen in coral. The Gray-Scott model is highly effective for producing coral-like ridges and labyrinth patterns. It uses a variation of the general reaction-diffusion equation. Other common models include the classic Turing model, the Nagumo model for wave patterns, and Fokker-Planck models for probability transport. The computational workflow begins with reaction-diffusion simulation to compute the 2D/3D patterns. This is converted into elevation data via a height field, then meshed into a 3D printable geometry, exported as an STL file, and sent to large-scale bioprinters. Software used includes Python or MATLAB for solving equations, Blender or Grasshopper for modeling, and specialized slicer software for printing. For 3D printed coral scaffoldings, we select substrates that encourage biological adhesion, such as terracotta clay, limestone biocrete, geopolymer concrete, and calcium carbonate composites. The biomimetic benefits include a high surface area for microbiota, microcrevices for larval settlement sites, and hydrodynamic complexity that disperses nutrients effectively. Archreef's work provides a successful example of 3D printed coral reefs. They use brain coral-inspired terracotta tiles for restoration. This includes the tile detail, deployment in Hong Kong, and visible coral recolonization on the structures. The world's first terracotta coral tiles resulted from a collaboration between marine scientists from the Swire Institute of Marine Science and architects from Hong Kong University. This partnership exemplifies constructing innovative solutions to conserve vital marine habitats. WASP's 3D printed coral reef restoration provides another example of biomimetic design. Their modules, inspired by brain coral patterns, are created using 3D printing technology with sustainable materials like terracotta and clay, aiming to restore damaged reef ecosystems and boost biodiversity. Our proposed field experiment is set for the Gulf of Mannar over 3 to 5 years. We will compare standard geometric shapes and commercially available structures with our mathematically generated biomimetic modules. We will measure new polyp attachment, the return of species, symbiotic algae growth, and coastal protection from wave energy reduction. The restorative ecology benefits are substantial. Expected outcomes include accelerated coral colonization, rapid biodiversity restoration, significant coastal erosion reduction through wave dissipation, and immediate enhancement of reef habitat complexity. Globally, this technology supports reef restoration, marine aquaculture, and coastal protection by creating living biological barriers. Future research will focus on using machine learning to tune reaction-diffusion parameters to match specific coral species. We will couple computational fluid dynamics with pattern generation for local current regimes, develop printable inks derived from biological aragonite, and explore automated drone placement of pre-seeded modules. Reaction-diffusion systems offer biologically realistic geometry, enabling scalable 3D fabrication pipelines and providing massive ecological restoration potential for reef systems. These are the image sources used throughout this video. In conclusion, mathematical models like reaction-diffusion generate complex biomimetic geometries. 3D printing enables scalable fabrication with materials like ceramic. Artificial reefs act as scaffolds to restore marine biodiversity, and real-world pilots demonstrate successful ecological integration.